The overall goal of this protocol is to acquire the skills and knowledge that allows the purification of DNA from a band fragment cut from agarose gel, utilizing agarose fusion and purification columns.
A very common step in cloning experiments is the purification of DNA fragments before ligation. This means that the step before cloning a fragment of interest includes that the insert and vector will derive from restriction and the nuclease digests, and will be mixed with enzyme cells and other DNA fragments that are not needed for further steps. A simple solution is the purification of DNA of interest, which involves an agarose gel, electrophoresis for separation of the diverse fragments, followed by the extraction of the desired one. The following purification method differs on which of the existing is to be completed.
These methods can be made in an elution from glass or silica particles, DEA as well as membrane, crush and soak method, electrolytion and respective commercial purification kits. Those selected methods will depend mostly on what is available in the laboratory. The basic methods and kits use a catch-up agent, such as sodium iodide, that destabilize the agar gel. Then, DDI is bound to a substrate that differs according to the method, but could, for example, be an ionic resin. Then, is washed to remove the impurities, and finally, the DNA is eluted from the substrate.
When regarding columns, it is in essence a silica-type membrane with a spin column alongside a number of buffers and wash solutions that help bind, wash, and elute the DNA. The silica membrane binds the DNA molecules in the presence of high ionic salt buffers that permit hydrogen bond formations between the silica and the DNA. The DNA sample is bound as it passes through the column by centrifugation. Washes are needed to eliminate unwanted molecules and impurities, including nucleotides, proteins, and salts. Thus, a low ionic or low salt solution is used for DNA release, as it disrupts the hydrogen bonds previously formed that bound the DNA to the membrane. Elution is the most efficient in basic conditions, and warming the elution buffer and letting it sit on the membrane for as long as 5 minutes favors the release of more DNA molecules.
An agarose gel was prepared at a 0.8 concentration with gel rate 0.3x and TAE1x. The wells were of approximately 10 mm wide. The sample was prepared with 45 microliters of genomic DNA and PCR products in order to have enough nucleic acid for further purification. This gives a total of 90 microliters of DNA mixed with 18 microliters of loading buffer, and 100 microliters total volume were loaded into the gel. Two base pair markers were loaded flanking the diverse wells corresponding to different DNA samples. The gel was set at a 120 volts for 1 hour.
When the electrophoresis was completed, the gel was visualized in a dark room on a transilluminator, and a scalpel blade was used in order to cut around the band of interest without reaching the bottom of the gel. The gel was then taken from the UV light and properly excised with as little agarose as possible and placed in a microfuge tube.
The weight of DNA was calculated, and an L3 buffer was added in 3 times its weight in milliliters, then incubated at 50 degrees, mixing every 3 minutes for 10 minutes. This was then transferred to the extraction column with a total volume of 722 microliters, being that the capacity of the utilized column was 850. The column placed in the microfuge tube was centrifuged at 12,000 gravitational forces for 1 minute, and the liquid discarded.
500 microliters of wash buffer were added and then centrifugated again at same condition but for 10 minutes. The liquid was discarded and the column was transferred to a new microfood tube. 50 microliters of lotion buffer were added and incubated at room temperature for 1 minute, then centrifugated at same condition for 1 minute as the final DNA elution step.
A 1/100 dilution was prepared using 10 microliters of the purified DNA and 990 microliters of distilled water. The absorbance was read at 260 and 280 nanometers, as well as the concentration and the absorbance 260/280 index.
The spectrophotometer reading gave us a total concentration of 1.1648 nanograms per milliliter or microliter, that multiplied by the 90 microliters used gives us a total of 104.832 nanograms of DNA. The A260-A280 index obtained was 1.3112, whereas it should have been 1.722 for an acceptable purity.
A purification process requires analysis in order to determine whether or not what was carried out was done so successfully, and the most used methods for this are using the spectrophotometer to determine the concentration as well as purity index by absorbance, as well as electrophoresis that will allow the observation of the fragment of interest as it is desired, thus proving the spectrophotometer readings were indeed correct. Another factor that is relevant is reading the absorbance in order to determine the concentration of DNA in a sample prior to purification, and afterwards in order to determine the yield in percentage, but in this case this was not done.
Indeed, it is important to mention some crucial parts of the protocol for completing a favorable purification. Agarose gel is used at 0.8% because this concentration allows the gel to be gotten rich for a better purification. Furthermore, Thai is used to oppose TBE, for it has a lesser concentration of salts, so borate carryover could affect the ligation. It is also an enzyme inhibitor.
DNA was visualized in the transilluminator very quickly in order to excise the band, and the adequate protection was always used due to the fact that UV light is dangerous. The quickness is because UV mutagenizes the DNA at a measurable rate, and this would interfere with downstream processes. Finally, the buffer used for solubilizing the gel-encased DNA has a specific pH which is maintained for optimal DNA binding. Acidic pH usually enhances DNA absorption to the silica membrane. This favors the most accurate elution step for a better purification.
The extraction of DNA fragments from agarose gel is needed constantly for possible downstream applications involving the fragment of interest. The correct manipulation and completion of the steps involved leads to a desired purity that will allow further protocols to be achievable. Depending on the method used for purification, there are variations of the basic binding, washing, and eluting system that is most commonly chosen upon reagent and equipment availability in the laboratory.